Good afternoon, students. This is part 1 of your exam for your Chapter 1 review on the study of human development, biological foundations, and the prenatal environment of the newborn. So we first start talking about how to define human development, and recall it is defined as how and why people change and how they remain the same over time. Recall Baltes says that lifespan development covers many different areas and that it is multidirectionality. It also examines how and why people are both unique, different, or how they are similar to each other. It is a multidisciplinary science based on theories and several research studies which we will discuss. So, we talked about the nature-nurture debate. Remember, the nature-nurture debate is heredity versus the environment, how each of those influence us in our growth and development as we grow and change throughout the lifespan. The nature in the debate refers to our heredity. How much does our heredity influence our growth and development? And the nurture is the environment. How much does our environment influence us as we grow and develop? Make sure that you know the difference between those two concepts. We talked about research theories. Recall that a theory is an organized set of ideas that is designed to explain development. What do we want to study? Something that we have an idea about that we are interested in. It is essential for developing predictions about behavior. We can predict behavior based on certain variables in the environment. Also, predictions result in research that helps to support or clarify the theory. We conduct research to form questions and answer them throughout controlled experiments. And remember, there are no one true theory about human development because we discussed several theorists who had different ideas about growth and development. So let's talk about psychodynamic theory. It goes all the way back to Sigmund Freud's theory that personality emerges from conflicts in our childhood. Remember, he believed that it was the unconscious, those things that we hide or repress that causes us psychological discomfort. Recall Erickson's theory was the first one about lifespan views on psychosocial development. The psychodynamic perspective emphasizes that the trends to adulthood is difficult because it has a lot of challenges as we grow and develop. Another area we talked about was behaviorism, which is based on a theory and perspective that in psychology that holds that conclusions about human development should be based on controlled observations of overt behavior or observable traits rather than on speculating about unconscious or other unobservable phenomena. And it's also the basis for learning. Key figures we discussed about in behaviorism was 1. John B. Watson, who discovered classical conditioning, that learning is based on association of certain stimuli, and B.F. Skinner, who said that behavior was based on consequences, rewards, punishments, and reinforcement. Skinner's theory we discussed in great detail was operant conditioning, which is what he said that the consequences of a behavior is determined whether it will be repeated or not. What we do. A positive or negative reinforcement increases the chance that a behavior will be repeated. Example, giving a child $5 for every 8 on the report card is a positive reinforcement. Negative reinforcement, taking away that video game because the grades will not improve. Punishment decreases the chance of the behavior will be repeated. Getting a traffic ticket for speeding or breaking the speed laws. So make sure that you understand the differences between positive and negative reinforcement, positive and negative punishment. We also talked about Piaget's cognitive developmental theory. His theory focused on thinking that development occurs in a sequence of stages, and his main emphasis was on the biological and social forces, less on psychological and little on the life cycle. And he said that his stages occur in 4 steps. The sensory-motor stage, which is age 0 to 2 years, where the infant's knowledge of the world is based on senses and motor skills. The example, out of sight and out of mind. The pre-operational stage, ages 2 to 6 years. The child learns how to use symbols, such as words and numbers, to represent aspects of the world, but relates to the world only through his or her own perspective. Concrete operations, 7 years to early adolescence. The child understands and applies logical operations to experiences provided that they are focused on the here and now, the present, rather than the past. And finally, his formal operation stage. I believe that it goes adolescence and beyond. Adolescents or adults, they can think more abstractly and they can deal with more hypothetical situations and speculations about what may be possible. Younger children cannot do this. So make sure you understand Piaget's stages of development and examples we occurred in class. You will have a couple of case studies that you will have to identify. Psychosocial development, again, is the psychodynamic theory. Erik Erikson, and his main idea was that personality develops through a sequence of stages, and his main emphasis was more on the psychological, the social, and the life cycle forces. He placed less emphasis on biological as well. And his main thing, again, went back to the nature-nurture interaction within the environment and heredity. He said that there is a universal sequence, but individual differences are rare. All right, we're talking about measurement in human development. How do psychologists study psychological issues? And how do they collect data to conduct their studies? So recall, we talked about 2 different types. Systematic observation. The first type we talked about was naturalistic observation, where the researcher observes the participant or a group in their natural environment to study and collect data so that they can form a hypothesis about real life situations. They actually go and observe. The other type of systematic observation we talked about was structured observations, where researchers create situations likely to result in the type of behavior in which they are interested. Example, my supervisor comes into the classroom to observe my teaching methods and student responses. So make sure that you know the definitions between naturalistic observation and structured observations. Some other behavior measures that we talked about were self-reports, where they measure a certain construct, such as self-esteem. And they use questionnaires to do this. We talked about representative sampling in experiments, which the sample must reflect the characteristics of the population of interest that's being studied. Recall that the population is the group of people in which the researchers may be interested in. And from that population, they will take a small sample of people who are randomly assigned to participate in the study that can represent the population that's studied or generalize to the population. Reliability. Does this method consistently measure what is being studied? Example, if an exam is being given to a group of students, and if the same exam is given to the same students under the same ideal standards, are the scores consistent on the exams given 2 or 3 weeks later? Is it consistently measuring what it's supposed to measure over a certain interval? Validity. Does the exam accurately measure the variable of interest? Is personality measured? Are the questions directly toward collecting data about different constructs of personality? Am I measuring you on the objectives taught in class rather than some other information that we have not discussed? That's the difference between reliability, which is consistency, validity meaning accuracy. We talked about general research designs, correlational studies as well, which measures how scores on two variables are related, SAT, GPA. And they calculate a correlation coefficient, which is a number between the two variable scores. R is the correlation coefficient, which it can range from negative 1.00 and plus 1.00. The numbers represent the strength of the relationship, and it shows how much the scores of one variable can predict the other variable scores. In a correlational study, you cannot prove that it caused the behavior or the results or outcome. There are three types of correlations we talked about. Positive correlation: as one variable increases, the other variable is expected to increase. Example, GPA times been studying, or GPA and high grades on the exam. And then in a negative correlation, as one variable decreases, so does the other variable expect to decrease. Example, the number of hours spent practicing piano and the number of mistakes made during recital. Another type of research design we said were experimental studies, where they study the possible cause and effect relationship between two variables. Remember, correlation studies do not indicate cause, but only a prediction. But experimental studies can prove cause and effect, that one variable causes the outcome of the other. Independent variable: this is a variable that the researcher manipulates, the key factor being tested in the experiment. Dependent variable: the variable that is the behavior being observed in the experiment and is used to evaluate the impact of the independent variable on the dependent variable. Example, does sunlight causes plant growth? Plant growth would be the dependent variable. Sunlight would be the independent variable. So make sure that you understand how to determine what the dependent variable is and the independent variable is. What is a control group? It's the group that does not receive the treatment, while all other factors are being held constant. And the treatment group is the group that receives the treatment and are being studied and compared to the control group. So the control group can be considered the comparison group. Then we talked about qualitative studies, which involve where they go more in depth when they want to understand a phenomenon, and they more than likely do this in conjunction with survey studies. It's smaller and it's more focused on what they want to look for. We talked about longitudinal studies. We talked about cross-sectional studies. And you did assignments where you had to learn to determine which will be best done in longitudinal, cross-sectional, or sequential. Recall that a longitudinal study is where you observe or you test the same group of individuals at each of several time periods. You test the same individual or groups over a long period of time. That makes it longitudinal. However, there are some disadvantages to longitudinal studies. It's very expensive and it's very time consuming. It allows examination of discontinuity or continuity because the same people are tested across time. People can move, people can drop out of the study, and this will make it difficult for a longitudinal study to be accurate for the researcher. In contrast, our researchers also do cross-sectional studies where they observe or they test groups of people different ages at the same period of time. Here, the advantages here, more time effective and it's less expensive, but it cannot address the continuity or the discontinuity because each age group involves different people. And it allows for cohort effects. Differences may result from each group, makes it unique. So those are some of the disadvantages. So make sure you understand the difference and the advantages and disadvantages of longitudinal and cross-sectional studies for your exam. You will have to identify it. Also, we talked about sequential studies, which is a combination of cross-sectional and longitudinal designs. It's a more flexible way to collect information, and it avoids cohort effects of cross-sectional design. So what it does, it allows the researcher to look at survey data as well as categorical data. A meta-analysis is where you have multiple studies that examine the same research questions and variables. Example, studying drugs for clinical efficacy. More than one researcher can conduct research and collect results, and they should get the same results. In other words, these designs must be replicable. It also allows scientists to verify whether the same relationships among the variables are replicated across multiple studies, as we just said. That is a meta-analysis. We talked about research ethics, which is so very important, and you had an opportunity to examine the APA Codes of Ethics, Standard 8, where before any researcher can conduct experiments or research studies, they have to first obtain permission from the IRB or Internal Review Board to conduct human subjects research. But there are certain federal guidelines that the researcher must adhere to. First, they must be given informed consent. They must inform the participant everything about the nature of the research, everything that is involved with the research. They must avoid deception unless it's going to affect the outcome of the research or cause any kind of harm to the participant. Their confidentiality must be maintained. The researcher cannot discuss results of the data while being collected. They must minimize harm to participants physically, psychologically, or emotionally. They must allow participants to drop out of the study at any time. And they must debrief the participants at the conclusion of the study. They must give them the results of the study. And if there was any deception going on during the study, they must explain that concept and why. Finally, they must communicate the results. It becomes public knowledge. They must publish in a professional scientific peer-reviewed journal. And in order for them to publish in a journal, it has to be original research First, it has to be useful, and it must have been conducted well, following all these guidelines and scientific steps. And it must be a study that nobody else has done before in order for it to be published. Make sure you understand research ethics and everything that is involved. You have several questions on your exam about research ethics. Then we talked about the heredity and genetics biological components. Remember that the human egg contains 23 chromosomes from both the male and the female, and we have 23 from dad, 23 from mom, which makes a total of 46 chromosomes. And remember that the first 22 pairs of chromosomes are autosomes, and the 23rd pair of chromosomes are the sex chromosomes, which determines the child's sex, which is an XY chromosome pair. Now, in order for the child to be a baby girl, it must have 2 X chromosomes, 1 X chromosome from the mother and 1 X chromosome from the father. In order for it to be a male, you must have an X chromosome from the mother and X and a Y chromosome from the father, for a total of 46 chromosomes. That the human egg unites, it results in what is called a zygote that contains the complete set of chromosomes. So make sure you understand the chromosome pair. Genes are a group of compounds that provides a specific set of biochemical instructions that make up our DNA. And then within that DNA produces our genotype, which is a complete set of genes that we inherited from the parent. Genes is what we inherit, which helps determines our phenotype, which is a combination of genotype and environmental influences that result in the expression of traits or the physical or outward appearance, such as our behavior, our physical makeup, our psychological components, and such as our psychological, how we feel at characteristics, and the phenotype, outward appearance, blue eyes, brown hair, our height, and that kind of thing. So make sure you understand the differences in genotype and phenotype on your exam. We'll talk about dominant and recessive genes. The dominant genes is where certain genes are dominant over other genes. The dominant gene is usually the stronger gene, and the recessive gene is usually the weaker gene. Examples for a dominant gene pair: the 2 uppercase letters SS, or uppercase letter and lowercase letter. The uppercase dominant gene always take place. The recessive gene pair is the weaker gene. And how do you identify the recessive gene pairs? The 2 lowercase letters, such as SS. Incomplete dominance is what leads to a phenotype between the genotype, which is associated with either the dominant or the recessive gene pair. Example, if a person has a mild form of sickle cell trait that appears under physical exertion. We'll talk about the sickle cell in a few minutes. All right, and single gene inheritance. Heterozygous, when we speak of heterozygous, we are always talking about difference. A heterozygous father and a heterozygous mother might have a healthy child, or a child can have some sickle cell trait, or a child can have sickle cell disease. Recall that sickle cell is a genetic disorder linked to the sex chromosome. The mother and or the father can be a carrier of the disease, have the trait, but not have the disease. The child would have or could have the sickle cell disease. Other genetic disorders that you need to be aware of, PKU, which is a recessive gene, and make sure that you study sickle cell. Recall that sickle cell is where the red blood cell sickles or is bow-shaped. And of course we have dominant genes. Huntington disease, recall, attacks the nervous system. So make sure that you study what Huntington's disease is. Other abnormal chromosomes or damaged chromosomes can result in abnormal development, like Down syndrome, which the child may have 47 chromosomes or an extra chromosome. And we often call it trisomy 21, which is 3 Y chromosomes. Turner syndrome and Klinefelter syndrome. Make sure you understand which of these diseases, one specifically affects females and one of these diseases specifically affect males. So make sure that you understand those concepts. We talked about twin studies, and you are in the process of completing a research review article on several twin studies. Recall that dizygotic twins are called fraternal twins. Fraternal twins result from 2 different eggs and fertilized by 2 different sperm. And they are no more genetically related or similar than regular siblings. They're more like sister and brother. They could be sister and brother, 2 sisters, or 2 brothers. Monozygotic twins are called identical twins, which result from the union on 1 egg and 1 sperm that splits into soon after conception. Monozygotic, called identical, they result from the union of 1 egg and 1 sperm. Dizygotic or fraternal, and they result from 2 different eggs fertilized by 2 different sperm. And the monozygotic or the identical twins usually share much of the same environmental experiences, and they each have 50% of the genes from both parents because they are identical. Scientists compare identical and fraternal twins to measure the influence on heredity, whereas adoption studies compare adopted children to their biological parents' heredity and adoptive parents' environment. So this goes back to the nature-nurture environment, and as you complete your research review on your twin studies, then this is where you go back to the nurture-nature debate on those questions. And of course, they can do DNA sampling to see if the genotype is associated with behavior and phenotypes. Periods of prenatal development. Remember that prenatal development begins where? At conception. And it takes an average of 38 weeks or periods and is divided into 3 periods. So the first period is the zygotic period or the zygote, which lasts from weeks 1 and 2, and it travels down the fallopian tube and is implanted into the uterine wall. That is the first period, the zygote or the zygotic period. Then period 2 is the embryonic period, which lasts from weeks 3 to 8. This is where the body structures, all of the internal organs, the 3 layers of the skin develops, the amniotic sac fills with fluid, the umbilical cord connects to the embryo to the placenta, and this is where the exchange of nutrients, oxygen, vitamins, and waste products and the mother and embryo are stored. And even during this period is where they have the cephalocaudal principle, where the head develops before the rest of the body, even in utero. Recall those pictures we looked at, and you will see a picture in a minute. The proximal distal principle is where the growth of parts near the center of the body occurs before those that are more distant. So make sure you can identify the cephalocaudal principle and the proximal distal principle. The final period, or the third period of development, is the fetal period, which lasts from 9 to 38 weeks immediately after the embryonic period. This is where the rapid development of major organs are taking place and develops. The heart, the lungs. Movement is felt by the mother during this time, and the mother gains rapid weight at this time. We also talked about the age of viability. At what approximately how many weeks can the baby survive outside the womb if born? Age 22 to 24 weeks. Pregnancy, they can survive outside of the womb. If born less than 22, 24 weeks, the fetus has less chance of survival. So be sure that you understand the 3 periods of prenatal development and that you understand what the age of viability means. Recall that we talked about several risk factors during pregnancy, that during prenatal development, the mother has to take care of herself. Remember, proper nutrition is so very important, and that she should increase her calories and increase her nutrient intake during this time. So diet is very important. Stress is also very important. It's important that the mother try to avoid stressful situations as much as possible, because it can cause more harm when it's expressed in early pregnancy. Also, the mother's age as well as the father's age is very important during pregnancy. Prenatal development is most likely to proceed normally when women are between 20 and 35 years of age. Beyond that period of time, age period, then if the mother becomes pregnant, then there's a greater risk that the child could be born with all types of disabilities and genetic conditions. So this is the end of part 1 of your study. Part 2 coming very shortly.